All right, so I'm Alexis Battle. I'm faculty at Johns Hopkins, and my lab primarily uses machine learning methods to predict the effects of genetic variation in the human genome. And today I'll be talking about just a small component of my lab has been working on, but I think it's one of the biggest sort of open, challenging problems in our area, and something that I'm happy to tell you about. So I'll be talking about the analysis of rare genetic variants. So the reason it's very important to understand the impact of rare genetic variants is that rare variants are really abundant in human genomes. If you look at any given person and you get whole genome sequencing for that individual, the number of rare variants that you'll find, and here I'm defining rare as less than 1% minor allele frequency, you find tens of thousands of them. So the median you find is about 50,000 rare variants per person. So this is a lot of variants that we currently don't really know how to interpret. And further, both from sort of a theoretical perspective based on principles of natural selection and also based on empirical observations, rare variants are much more enriched for known deleterious classes of variation than common variants. So here, I'm showing a common score that we use of deleteriousness called the CAD score compared to the derived allele frequency. And you can see that the CAD score is generally predicting much more deleteriousness for rare variants than for common variants. So essentially we have, for any given person, something like 50,000 rare variants that we would like to be able to interpret if we really care about what's potentially going wrong in that person's cells and biology. And these are really, really difficult to predict the consequences of, especially for non-coding and regulatory variants, which is one of the primary focuses of my lab. Huge number of variants. We have very little ability to say anything about them. So what kind of progress can we make? So there's lots of different goals that you might have in the analysis of rare variation, and I'll just briefly mention them, although I only focus on a few. One is, of course, understanding human ancestry and population genetics. This is not the specialty of my lab. Another is understanding the genetic architecture of disease. So asking for any given disease or for complex traits, how much do rare variants in aggregate really contribute to the heritability of this disease versus common variants? This is important both for understanding particular genetic variants that affect disease, but also for understanding what sorts of studies should we design to study that disease. And of course, we'd like to identify the individual variants that increase risk. A thing that I spend a lot of time studying is the genetics of gene expression. So understanding, similar to the heritability question for complex traits, how much in aggregate do rare variants versus common variants contribute to the heritability of the expression of any given gene? And this is actually something that Noah, who's visiting here along with Ryan Hernandez at UCSF, has a recent paper about that you might want to check out. I'm not sure if he'll talk about it here. And also, of course, which are the specific rare variants that can affect gene expression. And if we identify particular rare variants that affect disease risk, this type of analysis can potentially help us understand the mechanisms through which a rare variant affects disease. And finally, of course, if we want to move towards sort of the full beauty of personal genomics, at some point we're going to have to be able to say something about these 50,000 variants per person that we get, right? So the motivation, this is very basic, but just to kind of go through the example, in a standard association test, whether this is a genome-wide association test for a disease trait or an EQTL study for gene expression, we're really just going through variant by variant and looking at some sort of association statistic between the genotype at a particular variant and our trait of interest. And we get a p-value or an odds ratio out of this from a simple association test. So that works really nicely for common variants, but if we apply most standard approaches to the analysis of a rare variant, perhaps we have a data set where only a single individual has a particular allele at the locus we're looking at, we're not going to get anything sensible out of that. So standard approaches generally will fail due to power, or if you have a particular method that's not well suited for outliers, it might actually give you a false positive for a rare variant. So this is not what we want to do. But as I said, many interesting variants in your genome, and actually many of those that might actually be most important for disease risk are going to be rare. So we can't perform these standard association methods in either disease or expression studies. So what do? And we think, just to sort of preview this, we're going to have to generalize from other variants we have seen before. So this is kind of a perfect opportunity for machine learning. So I'll go through a tiny bit of background, primarily focused on protein coding variation to give you a sense of the types of methods people have used in rare variant analysis, although again, protein coding variation is not my primary focus. So exome sequencing, really focusing on the exome and mostly protein coding variants, remains really popular in clinical settings and for the analysis of rare variation. So why is this? It's primarily because we don't know what to say about the rest of it. So protein coding variation is much more readily interpretable just using the genetic code. We know how to identify non-synonymous and loss-of-function variants. We also think that coding variation is disproportionately deleterious. So if we're going to look somewhere, this is a good place to start. There are also lots of scores available beyond just sort of loss of function and non-synonymous that can, in aggregate, using all the properties of a particular variant, predict the probability that a particular rare variant or any variant might affect protein structure or function, such as polyphen and SIFT. So, a lot of clinical evaluation and clinical genomics really does remain sort of semi-manual, bringing automatic annotation of variants along with knowledge of, the variants themselves, knowledge of the disease, the genes and the pathways involved in the disease, the mode of inheritance of the disease. All of this kind of comes together to diagnose a patient. So if you take 50,000 variants that have very little annotation attached to them and throw them at a clinical geneticist, they're not going to be able to do a lot with that. So the exome really remains very popular in these settings. Things have kind of moved beyond that, so our ability to assess the impact of a protein coding variant has been helped by very large data sets, such as the aggregation performed by EXAC that's collected over 60,000 healthy exomes. So this allows us to get large-scale comparison. For a patient that you have, you can take a given variant that's protein coding or in the exome. You can get a precise estimate of its frequency in a healthy population. You can also get estimates of, let's say you know that this variant is very likely to affect protein function, but you don't know if that gene really matters, right? And you can actually use this estimate of the probability of loss of function intolerance that they've estimated for every gene to get an assessment of whether that gene is likely to actually be disease-causing and therefore not found in a healthy population. Going beyond that, if we really want to start doing something like association testing, methods have been developed for quite a number of years now. So the easiest idea is perhaps this burden testing notion, where we can aggregate rare variants affecting the same gene into a unified association test. So I may have a data set where none of my individuals have the same rare variant in a given gene, but many of them do have some rare variant in a given gene. So rather than associating a given genotype, a specific locus with phenotype, I can create an aggregate score where I, for instance, count up the number of variants that each individual has in this particular gene, in gene A, and get this count for every patient, and then associate that count with the phenotype directly. And you can do slightly smarter things, such as only counting the non-synonymous variants or things like that. And you can get an association test out of this that's then a gene-level association test and tells you how important rare variants in this gene are to your phenotype. Going sort of beyond that, there's been application of mixed models that also aggregate effects across a region and offer a number of advantages. So here they're really just modeling the phenotype Y as a linear model where each rare variant or each even rare and common variant in this vector G contributes something according to an effect size beta. And they can also take into account in the same model covariates. So that offers kind of one immediate advantage over just the simple burden test idea. And then rather than test every variant at the same time, they're testing a null hypothesis that all of the rare variants have an effect size of 0, and they test that jointly using this mixed model approach. So this is another way of doing gene-based or region-based testing directly from rare variants, and this approach offers a few advantages, such as I said, incorporating covariates directly. You can also actually re-weight your variants, so you don't have to count every variant equally, so you can weight them according to SIFT or polyphen, or their minor allele frequency, anything that you prefer. And you can even do things that are fancier, such as incorporate non-additive effects if you think that that's important for your analysis. So this kind of gives you a very brief overview of standard methods for rare variant analysis, primarily focusing on protein coding. But I want to move on now to talk about my focus, which is rare regulatory variants. So most rare variation is non-coding, so it's almost all 50,000 of them that are going to be non-coding. And can we interpret and use these in any way? And I'm just going to state, and you'll hopefully believe me, that our current approaches are very, very limited in their ability to predict the consequences of rare regulatory variation. So what are some of the difficulties in then using these in any of our analysis? There's many more of them. It's unclear, for instance, if we wanted to apply a burden test or a region-based test, how to even define the regions for aggregation. And one of the main problems is the annotation quality. So there are lots of functional annotations available for regulatory elements now, but each one is only very, very weakly predictive of impact. So this limits the applicability of methods like SCAT and the mixed models and also the burden test methods where we have to define a region. And if we have a lot of variants, many of which are benign or have very, very small effects, it's simply not going to give us good results. And further, it's very, very difficult, of course, to identify individual functional rare regulatory variants. So what sorts of features are available now? So most of you have probably heard a lot about ENCODE and epigenomic data coming from Roadmap and others that allow us to identify regulatory elements such as promoters and enhancers, often even in a tissue-specific or cell-type-specific way. We have conservation scores. We know transcription factor binding sites. We know all sorts of things about the genome. So we know a lot. But each one of these features individually is not particularly predictive of any sort of functional impact for an individual variant. But there are many methods that take these types of scores and try to aggregate them to make some kind of prediction about whether a regulatory or whether an arbitrary variant will have function. So lots of these have been developed in recent years. So these include CAD, which I've already mentioned, Guava, Basset, DeepSea, that are essentially all supervised methods. So they collect some data set of genetic sequence or variation, and they have features such as these I've described and more being developed all the time. And they treat each of those as a feature of a variant, and they simply try to predict whether a given variant then will be deleterious or have some functional consequence. But they need a labeled training set to do so. So all of these methods do rely on supervised machine learning approaches, ranging from something as simple as logistic regression or SVMs all the way to deep learning methods. But relying on supervised training means that you need some training data. And the real problem is that a large and unbiased source of variant labels for non-coding and regulatory variants is not really available. So each of these uses something different. So for instance, Guava is trained based on known pathogenic variants from ClinVar, for example. CAD uses an estimate of deleteriousness based on principles of selection, but none of them come without their biases. The Guava, for instance, the ClinVar set is quite small. Others are trained directly on functional data, such as chromatin, state, and others, but essentially getting a large and very unbiased source of variant labels is not obvious. So because we don't know much about which regulatory variants are actually functional, because we're so limited currently, it is very hard for us to construct a training set that doesn't come with some sort of bias. So again, I'm not going to provide a ton of background for this, but overall, performance of these methods, while I think very promising, remains insufficient for some applications, including sort of the ultimate goals of clinical genomics. So we're not going to improve directly on methods such as this. In fact, we're going to use all of these methods for further analysis. So the question that we're going to ask is, what can we do beyond this? So if we're now able to supplement whole genome sequencing in either a clinical or research setting with additional molecular phenotypes from the same individual, how much more information can we get, and how much will that help us? So for example, what can we gain by getting matched RNA-seq and whole genome sequencing from the same person? And how do we really quantify and integrate those signals together? So we really focus on the use of probabilistic graphical models, as David Knowles introduced to you yesterday, to sort of encode relationships between the different available measures and hopefully be able to give really well-interpretable and principled posteriors of functionality of a variant given any available data. So that's the approach that we're going to take, but I'll go through it a little bit more carefully. So I'll give just a little bit of background first in terms of the basic idea that we use before we get to the machine learning aspect. The hypothesis is simply that regardless of anything else, a functional variant that causes disease or causes some sort of deleterious effect to the organism very likely causes some kind of disruption at a cellular level. And so given the hypothesis that many of us have that most non-coding variation acts through gene regulation, extreme expression levels, so seeing that somebody displays a very extreme level of expression for a particular gene may be able to inform the effects of regulatory variation, even for very, very rare variants. So the simplest approach that we're going to start with is just identifying who are gene expression outliers. So this is for a given gene, one gene at a time. We could take a population of individuals and look at just the distribution of expression levels that those individuals show. And then for any given person, if we get RNA-seq or gene expression measurements, we can assess whether or not that person is an outlier just using a z-score. So we took this basic approach and applied it in the GTEx data that we've been working with for a long time. So GTEx does in fact have matched whole genome sequencing and gene expression data. And here, one of the advantages is that we get gene expression in multiple samples for the same person across a number of tissues. So we have here 116 people of European ancestry, and each of them has some set among the 44 tissues measured with RNA-seq in addition. So that does help us. So here, we're also going to consider looking at individuals who are outliers across multiple samples, simply using a median z-score. And as you'll see, multiple samples does provide us much more robust evidence, given all of the noise and bias that we see in RNA-seq. Seeing that somebody is an outlier across multiple samples is much more informative than seeing they're an outlier in a single sample. So the first question is simply, are expression outliers informative of rare variation? And it turns out that the answer is yes. So individuals that have rare variants, rare genetic variants near a gene are enriched for having extreme expression levels. So this first plot simply shows variants bucketed by minor allele frequency and the enrichment of extreme expression levels for those individuals. And as you can see, only the rare variants are really strongly enriched for displaying extreme expression. You can also see that holds strongly as we increase the threshold for what we consider to be an outlier. So if we have a z-score of 1, you see some enrichment, 2, greater, et cetera. So you can see that it sort of increases with the strength of how strongly you are an outlier. And then the two sets of dots here are multi-tissue outliers. So that's taking the median across the tissues available for that person versus single tissue outliers. So there are error bars on the single tissue dots. So they are significantly enriched, but with much greater enrichment than the multi-tissue outliers. And to give kind of an overview of what types of variants we're seeing near gene expression outliers, this splits it up. So the bottom bar is individuals that are not outliers. The middle bar is individuals who are overexpression outliers, and the top bar is individuals who are underexpression outliers. So you can see, first of all, that individuals who are outliers do have more rare genetic variants near the corresponding gene, and they tend to come from categories that are much more deleterious. Yes? Yeah, so here we're saying within 10 kb, although we've done this analysis with varying distances. I would say using our current analysis, we do not see strong enrichment once you get very distant from the gene. So we're really focusing on those that are quite close. So that's still a limitation of the analysis now. But yeah, 10 kb. Okay, so this kind of gives you basic evidence that extreme gene expression can be informative of the function of rare genetic variants. But we want to take that further and really see how close we can get to prioritizing regulatory variants from full individual genome sequences. So we'd like to be able to take an arbitrary variant from a given genome and give you an estimate or a posterior of the probability that that variant has a regulatory consequence. So again, we can apply any of the methods that I mentioned, CAD, Guava, et cetera, to get a score based on whole genome sequencing. So we could, for instance, just take a single nucleotide variant in a non-coding region, look at all of the features we have available from different sources such as Roadmap and others, aggregate those either using an existing method or a new machine learning approach, and estimate the probability of impact. And the question that I'm going to be asking starts off very simple, which is how much better of an estimate can I get if I observe RNA sequencing for nearby genes in the same individual. So for example, I may have a moderate belief that a variant has a functional consequence based on regulatory element annotations, but if I then see that that individual displays extremely low expression of the nearby gene compared to the population, that should increase my confidence that this variant is actually doing something quite a lot. Conversely, if I see that expression of all nearby genes looks totally normal, I may believe that that variant is actually not doing much of anything. So using the tools of probabilistic modeling, we created a method called RIVER. This is RNA-informed variant effect on regulation, but you just remember RIVER. And it starts off, if you look at the top half of the model, I've written it out so that it looks just like the methods such as CAD and others that I introduced at the beginning. We're going to start with annotations of whole genome sequencing for a given variant, roadmap, conservation, et cetera. And from that, we'd like to predict whether a given variant has a functional regulatory effect. Now, the thing that we're going to add to this is, of course, that we've observed gene expression for nearby genes. And here we're just simply going to treat this as a binary variable of whether that individual is an outlier for expression of a nearby gene. Now, the thing that makes this slightly tricky and interesting is that we do not require any labeling at all. So we're actually going to never observe the functional status of any of the variants in our training or testing. So no labeled variants are needed for training at all. And yet, once we train this model fully, we can use it to then compute posteriors of this latent variable, whether or not a given variant has a functional regulatory effect, given any evidence. So we can take a new patient where we have whole genome sequencing and RNA-seq and compute the posterior, or we could take an individual for whom we have only whole genome sequencing or only RNA-seq, and we can compute the posterior that a given variant has a regulatory effect directly from this. So just to give a little bit more detail, the model itself really is built on very simple tools. So if you look at the top half of the model, it is essentially just logistic regression, with the only difference being that this is an unobserved latent variable. So that really is just logistic regression in the top half. The bottom half is essentially naive Bayes. It's just a categorical variable based on whether or not the variant is functional. So you have some probability of being an outlier if there's a functional variant, and some probability of being an outlier if there's not a functional variant. So it's a very simple probabilistic graphical model. The only complication, of course, is that there is this latent variable. So we test every gene individually and every subject or patient individually, which means that this really is the model. There are no cycles in the model at all. So we can do optimization in this model using expectation maximization, and every time we need to do inference, whether during learning or for a new individual, inference is tractable, and we can just use exact methods. So we're just using expectation maximization here to deal with the latent variable. So it's trained entirely unsupervised, but it is a simple model, and we kind of expected to get out of it that the model would ideally pick up identifying patterns in genomic annotations that were predictive of a particular level of outlier expression for a given gene, and vice versa. We would be able to tell how strong of an outlier you need to be, or what patterns you need to see in gene expression that are then informative in terms of the likelihood of seeing particular genomic annotations. So it's really just picking up that, and it's just summarizing it in a convenient way for us so that we can then use the posterior on this latent variable to make predictions for new individuals. But it does do exactly what we hoped. So what I'm showing here, on the x-axis, every dot is an instance. The x-axis is the median z-score across tissues. The y-axis is the genomic annotation score, which you can think of as like the CAD score, but it's basically the score that you get from the top half of the model only. So it's only considering the genomic annotations. And the color of the dot gives you the strength of the posterior on the latent variable. So the darker blue dots are the ones that River thinks are likely to be functional. The light gray dots are the ones that it thinks are unlikely to be functional. And you can see that it's really simple. It's exactly what we want, that it thinks it's more likely to be functional when it sees both a high z-score and signal from the genomic annotations alone. And if you only have one of those two signals, it's less confident that you have a functional variant in your genome, right?
Your dots here are variants.
They're gene-patient or gene-subject pairs. So it's one person and one gene analyzed in every dot, yeah. So we do actually, that's a detail I didn't really go into, we do actually aggregate the variants near any given gene together. So it is kind of a region-based approach, although I will say in this case, almost all the time it was clearly one variant that was standing out as being functional, but the model itself does not distinguish which of the nearby variants for a given patient is relevant.
So the annotations, how are you matching, like, there are tissue-specific annotations, but you're looking at a median expression, so
yes.
Do you use all of them, or do you take them?
So we've done both, and I'm actually going to come back to tissue specificity at the end of the talk, assuming there's time. But we take just aggregate, like for instance, maxes or medians across different cell types, for instance, from roadmap or ENCODE. Although we have tried using the individual tissue or cell type level features also, we tend to find that in this version they don't work as well, but here we're really trying to predict median expression. So we'll come back to predicting tissue specificity at the end. Yeah.
How much is outlier detection tied to the cohort? So like if I have a sample that I have RNA-seq for that wasn't part of GTEST,
yes.
Like can I still detect if it was an outlier, or is that like really hard?
No, you can, and I will come back to that at the end also.
Okay.
Yeah.
So people have been using throughout this type of modeling where you have these functional annotations, and then there's like the causality status, and then there's a complex trait.
Mm-hmm.
And that have been like geared more towards common variants, because
Yeah.
that looking.
Yeah.
Is there a connection where you can expand this to where it's putting together rare variants as well as common variants?
Yes, definitely. And in fact, I think we should put together rare and common variants because sometimes if you have, for instance, two very large effect common variants, that can actually explain why you're an outlier. And if we don't take that into account, either we've worked on regressing it out ahead of time, but I think modeling it simultaneously would actually be better. And we have worked on common variant models that use annotations like this previously for EQTL prediction. So one thing that was nice, we see that the model is, although it's completely unsupervised, it is picking up the signal that we hoped it would pick up. And we do also see, for example, that pathogenic variants that are regulatory pathogenic variants do tend to score very highly according to the posterior. In fact, all of the ClinVar regulatory variants turned out to be in the top 0.1% according to river scores in our entire analysis of GTEx. These are actually absolute values. We take the median z-score and then the absolute value. Yeah, good question though. And it's relevant because maybe you noticed on a previous slide that actually underexpression outliers look like they're much more enriched for genetic effects than overexpression. So that is something that we could potentially improve. So this is not exactly a methodological component, but I really wanted to mention it because I think it's not just this problem, but lots of genomic problems where we don't have a good gold standard. So just like we had the issue that we don't have a large training set that has unbiased labels for functional variants, we don't have a nice unbiased source of those labels to evaluate our model either. And I think a lot of you are probably working with cases where you don't really have a perfect gold standard. So you're either relying on simulation or a correlated phenotype or something like that to evaluate. So that was a problem here for sure. And I'm only going to show you a couple of ways that we evaluated the model, but we have several if you look through our paper, both in the paper and the supplement, that we tried to look at it from different angles because we know we don't have this perfect gold standard. So what do? So one idea is that although we're looking at very rare variants, minor allele frequency less than 0.01, there are enough cases in GTEx where we have two individuals that have the same rare genetic variant that we can actually treat one as an observation and the other one as a label. So the idea here is that we take all of these, we call them N of 2 variants, and we remove them entirely from our training data. So we train the river model on everything but the N of 2 variants. Then we take one of our individuals, imagine a given variant, we take person 1, we use our trained model, and we compute the posterior for that person, the probability that that regulatory variant is functional given both their RNA sequencing and their whole genome sequencing data, and we compute this posterior as our river score. Now we take our second person, person 2, we haven't used them in training, and we haven't used them in prediction, and we treat whether or not that individual displays extreme expression levels, so their outlier status, we treat that as a noisy label. So that's going to be one of our labels that we evaluate. So using this evaluation, we can then compute an ROC curve using, for example, river in the blue, or we can restrict the model to only use genomic annotations. So here we're imagining that we did not have RNA sequencing data available and computing a posterior there. And you can see that the RNA sequencing information added to river gives us a big boost in performance. These AUCs are not huge, and that is something that was noted by our reviewers. It's like, well, they're just not that good. And I had two answers to that. One is we know the label is noisy, but frankly, the problem is hard. That doesn't mean that we can't make progress. It is just simply a very hard problem. But you can see kind of where river is making its improvements, which I think is important, is in this region of sort of very low false positive rate, it's able to find a few more true positives that it's very confident about compared to the genomic annotation model. So it's finding genomic variants that have reasonable looking genomic annotations, but then seeing the RNA-seq signal being extreme on top of that gives it enough confidence that it's able to predict those before it starts incurring a lot of false positives. So we evaluated this also against allele-specific expression and others. But one thing we wanted to look at was, again, prediction of pathogenic variants. And this, again, kind of echoes what I said about identifying a handful, but still an important handful of variants that River is very confident about in contrast to existing methods. So here we're looking at if we make predictions based on gene expression alone. So this is imagining you had RNA-seq but no annotations of the genome. You're able to get some differentiation between known pathogenic variants and background. If you look at genomic annotations alone, you're able to, again, get some prediction and separation between pathogenic variants and background. But if you apply River, which simply combines these two signals in a principled way, you get this set of variants. It's not perfect. It doesn't separate them perfectly. But you get this set of variants that River gives a very high confidence to in terms of being pathogenic compared to background, much more so than the individual data types alone. So, briefly mentioning, I know this is not a biological meeting, but we actually did go through and do some CRISPR validation. And this is something that I think is important. So even for the protein coding analysis that I mentioned at the beginning, it's rare to actually get all the way to C validation, right? And I think we don't have a great estimate of what our true false positive rate or false discovery rate is until we actually push towards validation of some of these variants. They are very hard to predict. But we were able to see that for some of our highest confidence variants, we do in fact see regulatory effects when we introduce them using CRISPR. So you guys can play around with River if you're interested. It's available on Bioconductor now. But what I want to talk about in the last part is just extension of this method. Using this framework of probabilistic modeling, we can add lots of other things to this model quite easily. So it's really easy to incorporate new genomic and epigenomic features, of course. It's actually quite straightforward to incorporate new types of molecular data on top of RNA sequencing. And we can start to make more context and tissue-specific predictions, as Anshul asked about. So one place that we're looking at this is actually alternative splicing, which David also brought up in his talk. So we know from recent studies that splicing can contribute a lot to disease risk as well. So we can take the River model, and instead of thinking about total expression, we'd like to think about extreme splicing. This is a slightly more complicated idea, however. So total expression either goes way up or way down. You sort of only go in two directions. But if you have J junctions in a gene, if you're an extreme individual in terms of your pattern of splicing, you can be extreme in any of 2 to the J possible directions, right? So it's not simple to assess whether you're actually an outlier and how significant that is. So how do we define an outlier over a space like this? So one of my students, Ben Strober, is working on this method where he takes a population of individuals, and we're actually using the leafcutter model that David talked about, and we are looking at essentially junction counts over a number of individuals here, and we then model this as a Dirichlet multinomial. So we learn from our population of healthy individuals a Dirichlet multinomial that describes what normal splicing looks like for a given gene or a given cluster. Then for any new individual that we have, we can take their junction counts and compute the Mahalanobis distance for that sample compared to the parameters that we learned from the population. So this is just, the Mahalanobis distance is just a multivariate generalization of how many standard deviations a given sample is away from the mean, taking into account the variance and covariance of that distribution. Now, the complication is in assessing significance. So we can straightforwardly learn these distributions from, for instance, the GTEx data or others, and we can compute this distance for any given individual. But the Mahalanobis distance for arbitrary distributions, such as the Dirichlet multinomial, does not have like a closed form statistical test that you can look up. So what we actually do is randomly sample 100,000 samples from our fitted Dirichlet multinomial for a given gene. We compute the distance for each one of those randomly generated samples. So this should be an empirical null. All of these samples were generated from our population distribution. And then we can use those as an empirical distribution to assess the abnormality of any given sample, and we can get an empirical p-value out of that. So that's what we've done, and we've applied this also to the GTEx data. And this gives you an example of an individual that was called as an outlier in GTEx muscle tissue. So you can see that they have this region where they seem to be retaining what is an intron in the normal population. Almost no one has real expression there. This individual displays a lot of expression in this supposedly intronic region. So this is somebody that we're calling a splicing outlier. So this is the kind of thing that we detect. And actually, this is yielding a lot greater enrichment of rare variants than anything that we saw for total expression. So if you look at the y-axis here, the enrichment, fold change that we see is greater than anything that we've seen for any annotation that we checked for total expression. So our splicing outliers that we're calling as outliers are very, very strongly enriched for rare variants likely contributing to splicing changes. So we can actually take this new definition of what is an outlier, based now on splicing instead of total expression, and I can plug it into River without changing anything at all in the core model. So we can just run that immediately. And in fact, again, this echoes that we see much greater enrichment for rare genetic variants using splicing. The river model performs much, much better in predicting outlier splicing than it did for expression. So this looks like a stronger genetic signal. Of course, this does not cover every variant we care about. It doesn't cover those that affect total expression, but when it is a variant that affects splicing, we seem to be able to pick it up really well, just using the river model as developed for total expression and this new definition of who is an outlier based on splicing. So it's a really easy change to the model. Another example, several students in Stephen Montgomery's lab are working on changing the river model so that instead of just predicting one output of total expression, it can predict expression in one context. It can predict changes in protein level based on mass spec or ribosomal footprinting. But this is a totally simple change to the model. It's just extending the naive Bayes model to have a few outputs instead of one output. There's almost no change at all to the code or to the model or the inference. It remains a very tractable inference problem. So one thing that we're working on that gets a little bit more complicated is trying to predict context-specific impact of regulatory variation, such as tissue specificity. So we know that many factors, environmental changes in tissue, can alter things like transcription factor abundance, epigenetic changes, and others that can modulate whether a given genetic variant even matters in these different contexts. So without going into much detail, we are extending the river model to capture this in a model called Watershed. So here, instead of a single latent variable that assesses whether a given variant is functional, we actually have one of these latent variables for every context that we're interested in. So in the context of GTEx, this would be, for example, the 43 different tissues that we have RNA sequencing for. Each one of them then is a parent of whether that tissue displays extreme expression levels or not. So there would be, T tissues that will have T expression measurements separately and T latent variables of whether or not that rare variant has an effect in that tissue. The genomic annotations for a given rare variant, of course, are shared between all of the tissues here. So, this model now does actually start to have loops in it, so inference is no longer tractable. We can still apply the EM-based method, but now every time we need inference, we actually use variational inference instead of exact inference. So it's not as computationally efficient, but the underlying model is very, very close to what we had with River, with just this expansion across a number of contexts or a number of tissues. So this is the kind of change that is straightforward conceptually to make once you start building on a model like River that tries to capture the interactions of the different types of measurements that you have available in order to make the prediction you're interested in. So if I have a couple more minutes, I don't know how much time I have exactly, but I will talk just very briefly about trying to take models like this and apply them in clinical cases. So we are working with people at Stanford, including Stephen Montgomery, to try to look to see if we can identify variants that might be disease-causing in rare disease samples where exome sequencing was previously done and a clear causal variant was not identified. So they thought maybe it's a regulatory variant. They had whole genome sequencing done, and they also had RNA sequencing done for these individuals. So we have now 12 rare disease samples and also family members for some number of them. And we'd like to take our rare disease sample with whole genome sequencing and RNA-seq and compare it to some population background. So this question was asked. This individual was not sequenced as part of GTEx. They were sequenced in a different lab under a different protocol. They probably only have blood. What do we do? So what we're trying to do is actually switch away from using GTEx. So GTEx was a really nice baseline for us for estimating the properties of these, gene expression outliers and rare variants in a very controlled setting. They're supposedly healthy, although they are post-mortem donors. They had consistent sample collection and processing, so there were a lot of advantages. But the limitations were, it's still relatively small. Even in sort of its final state, it's going to be about 1,000 individuals with whole genome sequencing. Not every tissue of interest is included. There's a limited sample size for some tissues. And the question is really, does it capture the full range of normal expression or normal splicing? And we don't know. So what we turn to is actually some of our colleagues at Johns Hopkins in Jeff Leake's lab, who have this resource called Recount2, where they've actually taken all the publicly available RNA sequencing samples, including GTEx, TCGA, and everything on SRA. And while they can't, control that it was actually sequenced in different labs, they can at least run it through uniform alignment and quantification pipelines. So they are more comparable than if you just took totally disparate RNA-seq data sets. So I encourage you all to check that out if that would be useful for you. But the question is, can we take a huge resource like this, where we have over, 60, I guess we have 70,000 now, and I think growing RNA-seq samples available through Recount. Can we model what normal gene splicing or normal expression should look like based on this RNA-seq data set? So one challenge is that if you guys have looked through SRA, it is poorly annotated, and tissue is actually unknown for most of these samples. So with Shannon Ellis, they've gone through and actually built a predictor that can take all of the samples in Recount and actually predict things like sex and tissue for all of these different samples. So we now have predicted tissue. So this is a lot of caveats that I've given, right? It's coming from different labs. We are not completely sure of the tissue in many cases. So can we use this as a baseline to assess new individuals? So we actually turned back to GTEx to evaluate this. So we took our GTEx individuals and looked again at enrichment of rare variants. Now when we assess the Dirichlet multinomial or the gene expression level profile that we want to build for the population, this Dirichlet multinomial is now learned from all the Recount 2 SRA samples for the tissue that you are trying to evaluate. And then we went back and for our GTEx individuals called outliers based on this Recount Dirichlet multinomial and looked at enrichment. And actually what we see is that depending on how you match the number of outliers you're comparing, if you look at p-value, actually Recount 2 gives you better enrichment than GTEx did. Now, I don't think this comes purely from having an increased sample size. I actually think some of the problems turn out to be an advantage. So the fact that I might have some incorrect tissues mixed in there, some disease individuals, just a bit more representation of the differences between sequencing protocols, et cetera. I actually think it gives us a better estimate of a broader range of what could be considered normal, and you're less likely to take your new individual that you've sequenced in some arbitrary way and maybe has some artifacts, et cetera. You're less likely to call that individual an outlier by comparing it to GTEx, this really nice, clean data set. So I think that's actually why it's helping. But we do see strong enrichment, so I do think it's evidence that we can use this as our baseline. And taking this then, we took all of our 12 individuals and we actually ran them through simply outlier calling. And while we don't have, I'm not going to share a disease diagnosis with you here. We are working on some individual variants that we think are promising. But I just want to point out what the difference would be to somebody looking at this in a clinical context. In this sample, we're seeing something more like 70,000 rare variants per individual. Even if you say, well, I'm really only going to look near a gene, within 1 kb of a gene for these individuals, we see on average 30,000 rare variants. This is not plausible to give to a clinician and say, here, what do you think? But if you take either of our methods, whether it's based on expression outlier calling or splicing outlier calling, we're now reducing it to a handful of plausible variants that somebody could actually look at and look at the genes and see if there is anything meaningful. So I think this is, while very, very, very far from perfect, really moving us towards what we'd like to do ultimately. So that's the idea here. So I'll go ahead and stop there, but the idea that I want to provide is that rare regulatory variant evaluation remains really challenging, but I think using new data types, including just RNA-seq in our same samples, provides much better evidence of variant impact than simply using genomic features alone or any aggregate score that's currently available. And using a relatively straightforward probabilistic model that's extensible to lots of different contexts and taking into account different information, it provides a basis for making these predictions and hopefully also evaluating them and getting some sense of your accuracy. So stop there and thank all of my collaborators, especially Stephen Montgomery's lab, Shin Lee, Joe Davis, Emily, and my students Young and Ben. So thank you.